With this video, our job is to look at the actions of antibodies and to understand how antibodies are constructed. And then we'll look at the time sequences of two different immune responses, a primary and a secondary immune response. And then last of all, we'll look at how we can classify immunity based on how we achieve immunity, how we get that immune response to occur. So we will start with antibodies. And there are 5 different classes of antibodies, also known as immunoglobulins. And they're denoted by letters. There's immunoglobulin A, immunoglobulin G, sometimes just referred to as IgA, IgG, or by their Greek letters. So, for instance, IgGs are sometimes referred to as gamma globulins because they are the G form of the immunoglobulin. So there's IgA, IgG, IgD, IgE, and IgM. So when you look at the structure of an antibody, there are 2 identical long chains. So I'm going to put a line here through the long chains, and they are identical to each other. And then 2 identical short chains. These are the short chains. And the long chains are sometimes referred to as heavy chains, while the short chains are referred to as light chains. And what they are is sequences of amino acids. So these are, peptides or small proteins. Each of these chains is. So each of the classes, the 5 classes, IgA through IgM, are distinguished because of the sequence of amino acids that are found in the constant regions of the heavy chain. So here in the heavy chain, this area that is green is called the constant region. And so that sequence is the same if it's IgA. There's a different sequence, but if it's an IgG, it also has, all of the IgGs have the identical constant regions in the heavy chains and so on. But the thing that makes each antibody specific is because there is also a variable region. So this distal end here of the heavy chain and the far end of the light chain both are variable. And it is because of the sequence that we find here in the variable region then that the antibody is capable of binding to a very specific antigen and no other antigen. So, just to recap, 5 classes. The classes are determined by the sequence of amino acids here in the heavy chain at the constant regions. And the light chain also has a constant region. We could point this out. And so everything that's here in this red circle is what determines which class of immunoglobulin that we would put this into. And the variable regions of the light and heavy chains, they are what allows each antibody to be specific. So let's take a look at each of the classes and see what they're used for in the body. So IgG, immunoglobulin G, the gamma globulins, these make up 80% of all the body fluids that are found, all the antibodies that are found in body fluids. And we see IgGs primarily in blood plasma and in the tissue fluids. They function generally to defend against bacteria, against viruses, and against toxins that may be introduced into the body. And they have the added benefit of being able to cross the placenta. So they make it from mama's blood, where baby doesn't have a chance to encounter antigens generally unless mother gets really sick. But all of the antibodies, the IgG antibodies that mother makes, though, are capable of slipping out of her bloodstream and at the placenta crossing over into baby's bloodstream. And so this is part of what provides passive immunity to newborns, is the fact that the placenta is permeable to the IgGs. IgA makes up the next largest abundance of antibodies. About 13% of all the immunoglobulins in body fluids are IgAs. And we see them primarily in exocrine gland secretions. So breast milk, tears, the nasal fluids, so the mucus fluids of the nose, in the gastric juices in bile, and we can also find them in urine. And they tend to primarily be used to defend it against bacterial and virus invasions of the body. So that's IgG and IgA. Excuse me, I got the coughs this evening. Immunoglobulin M is next in abundance, makes up about 6% of antibodies found in body fluids, and it's found primarily in blood plasma, and it defends against bacteria, against antigens that we might find in food, and also against the red blood cell antigens. Anti-A and anti-B are, those antibodies are IgM. IgD is found on the surface of B cells and plays a role in B cell activation. And so we're not going to dive too deeply into that, but that's where their function is. Immunoglobulin E is found in trace amounts in body fluids. It's also found in exocrine gland secretions and on mast cells out there in connective tissues. Remember, mast cells are the ones that promote inflammation when there's been damage to the body. So these IgEs play a role in promoting inflammation, and sometimes this inflammation can be so intense that tissue damage occurs. And so this is what we see when there's an allergic reaction and somebody is going into anaphylaxis. It is triggered in part by an excess of IgE around as part of an immune response. Okay, so when antibodies are produced by the plasma cells and released into body fluids, what is it that they do? How do they function to help provide immunity? So the first thing that they do is, and these are great big words, they cause agglutination and precipitation. Agglutination just means sticking together, and because antibodies are sometimes found in ways that allow them to bind multiple antigens, they can tie together a big old clump, and so we just have two antibodies here, but these two antibodies are able to bind together into a big clump 3 bacteria. Imagine if there were more antibodies kind of arranged like this around a molecule. Then we could bind even more of the bacteria together to make a large clump. That's agglutination, when we stick them together into large clumps. Now, once a bunch of antigen-bearing particles are agglutinated, this makes them insoluble and heavier than solutions, and so they come out of the solution. And that is referred to as precipitation, because they're no longer dissolved. If it's say a protein antigen, it's no longer dissolved in the fluids, but instead it is made insoluble. Both of these things, agglutination and precipitation, tend to make them more obvious to macrophages that are going to come along and clean things up. So agglutination and precipitation are very useful. The second effect is that the antibodies, if it's an antibody against a virus particle or against a bacterial toxin, if we make an antibody against these materials, neutralization can occur. And so here we see this is an exotoxin, so some sort of toxin that's been made by a bacteria, and it has these 2 binding sites right here and right here. So the red thing is the toxin, and it would normally bind here against the host cell. That's where it would normally go, causing its toxic effect. Let's see, the toxic effect inside of the cell. But antibodies can come and bind and cover over those binding sites, and it blocks their effect. It neutralizes the effect of the toxin. And the same if we have, say, here's a virus particle with its sticking out from it. It's got all of these receptors that would go and try to bind against a host cell. But if we can make antibodies that will come and cover over those binding sites, then now our toxoid, or our virus particle, is neutralized. Ha ha ha. So we've made it sad. Okay, so those are two effects of antibodies binding to an antigen: agglutination and precipitation, and neutralization. That's not all, though. If an antibody binds to an antigen, then it activates a whole nother layer of chemical defenses called complement. And there is some indication that activating complement may be the most important infection-fighting activity that an antibody can do inside the body. So when we activate complement, it comes and binds at the same locations as where the antibody has bound. And in the process, it helps to attract macrophages up to the particle that needs to be destroyed in order to rescue the body. It can also attract neutrophils, and they notice those chemicals and they direct toward them. We refer to that process of noticing and heading toward or away from a chemical as chemotaxis. And so macrophages and neutrophils are positively chemotactic. That means they're attracted to these complement proteins that are found that will come and kind of bind, kind of smear over the same area where the antibody is. They coat or cover over the antigen-antibody complex, so that's where the antibody has bound to the antigen. We refer to that coating as opsonization, so they opsonize the antigen-antibody complex, and that then also not only attracts, but it makes phagocytosis even easier. It helps serve as a great big flag saying, hey, come eat us right here, come munch down on this. Sometimes the complement proteins alone can cause lysis of the antigen-bearing particles. So if it's a bacteria, let's say, then the binding of complement there can also force that bacterial cell to lyse, to bust open. It can increase the agglutination effect of the antigen-bearing particles. So it works along with the agglutinating characteristics of antibody binding to cause even more agglutination to occur. It can actually change the structure of the viruses, mess with the proteins, and in the process render the virus harmless as we change those proteins. And then last of all, the complement proteins will help to stimulate inflammation to occur at the body site. And so, as we spoke before, this is all about helping to slow the spread of infections through the body. Okay, so lots of actions occur when we bind antibodies. We can agglutinate and precipitate. We can activate complement. Lots of things can go down as a result of antibodies' actions. But in the end, that is what will take down whatever it is that has invaded the body and needs to be removed in order to restore health. Okay, so what is the time series? How long does it take for this immune response to occur? And so we're going to look first at the primary immune response, which is the response on the very first time that we're exposed to an antigen. So initial exposure to an antigen, the first time, is what the primary immune response means. And in order to mount a primary immune response, we have to first activate the B cells, which also requires, in order to get them all activated, remember it requires activation of helper T cells as well. So we got to get all of that that activating going, and then it takes a while to get enough plasma cells made so that we can start really cranking out some antibodies. So all of that takes time. It's a slow process. And when we encounter an antigen for the first time, we don't get a whole lot of antibodies out of it. You know, a nice amount, but it's not an overwhelming amount of antibodies that get produced. So let's take a look here at the time course. Oh, and by the way, first, IgM is the first antibody to be produced, and it goes out into the lymph. And then the plasma cells shift over and begin producing IgGs. And at that point, antibodies then move into the blood. So we start our immune response in the lymph, and then it escalates to include the blood as well. And peak response occurs something on the order of 2 weeks, 14 days after exposure. So here's peak response, right? So here's first exposure, and we start noticing, hey, we got something in the body that doesn't belong. And it takes a while to even see the first antibodies being produced. But to get a whole bunch of antibodies, we have to get up here to about day 14. But with the production of antibodies, we can begin to fight off the infection. Now, generally, it doesn't prevent the disease, but it does help to fight the disease and to bring the disease under control. But as we destroy microbes, as we make more and more of these antibodies, because we've made plasma cells, we also make memory B cells. And some of these memory B cells can be super long-lived, 20 years or even more. Some of these memory B cells will hang out waiting to see if we're going to encounter that antigen again. If we do encounter the antigen, then we mount what is referred to as a secondary immune response. So it's a subsequent exposure to the antigen that we've already initiated a primary response to. Now I want you to do the comparison with me. So here is our primary response. You can see, again, day 14 is peak right about here, and notice we get about that much, so about halfway up the chart, we're looking at the amount of antibodies that are produced. And, after 20 days, we begin to see a decline. It's not a terribly steep decline, but over time, that primary response is going to get less and less. Let's look and see what happens when we have a secondary immune response. Notice that by day, let's see if this is about where we saw peak amount of production is right about there. Where does that look like? To me, that says at day 5 of a secondary immune response, we already reached the maximum that we reached in a primary immune response. And by day day 7 or 8, somewhere in there, so halfway to day 10, we hit the peak response. And it is something on the order of twice as much that peak response is in the number of antibodies that are produced. So, and then that response stays high for months. So for a very long time, we will see a high antibody count in the blood once we discover a secondary infection of the same pathogen. Why does it go so much more quickly? Well, first, because we have memory cells. These memory cells, when they encounter the antigen, rather than having to go through all of the steps of activation, they can go straight to making plasma cells. And so they immediately divide, and their offspring cells divide. And so we see this proliferation of plasma cells that go about making the antibodies. And so we get a very rapid and very intense response. And the good news for us is that rapid and intense response prevents the onset of disease. So rather than fighting the disease, like we saw in the primary response, with a secondary response, we may be able to completely prevent the disease from ever even being able to take hold. So before we even know we've been infected, our body has already defended us against it. So the other thing that helps to maintain a long-term supply of memory cells, remember they can last 20 years, sometimes longer, but we have a way of like constantly kind of reminding the body that, hey, we encountered some bad juju in the past and we want to be ready for it just in case. And so there are cells called follicular dendritic cells. They're found in lymph nodes, and they can aid memory by slowly leaking out antigens over time. And so we get this kind of low-grade little secondary response that helps to maintain an active, a healthy memory B cell population, and in that way provides us long-term immunity. So that's how we get immunity to many of what we think of as childhood diseases: chickenpox, measles, mumps, rubella. Those are all viral infections that we can develop long-term immunity to, thanks to the production of memory B cells and the action of the follicular dendritic cells that help to keep us constantly being on the alert, having sufficient memory B cells around that we can mount a secondary shock and awe type of immune response. Okay, so to wrap up then, let's talk a little bit about describing how immunity is gained. And so if we get exposed to, so what we're going to look at first is active immunity, so what we call active immunity. Active immunity occurs once we are exposed to an antigen and we go through a primary immune response to that antigen. And we have a population of memory B cells. That is how we get active immunity. And there are two ways to get active immunity. It could be naturally acquired. So that's, your kid came home from school with a nasty, viral infection. They passed it on to you, and you got sick and then recovered from it. That's natural acquisition of active immunity. But we can also induce active immunity, and that's what we do when we give people vaccines. We're administering either a killed or an attenuated version of the antigen-bearing particle. So it's a bacteria or a virus that's either completely dead. So what we're getting is dead cells that have the antigens on them, so that we notice those antigens and mount a primary response against them. Or they may be attenuated viruses or bacteria, where they are so weak that they cannot cause actual infection, but they are able to stimulate that primary immune response. We can also, if we're trying to get a immunity or vaccination to a toxin, we can take that toxin and convert it into what's called a toxoid. It's a non-toxic version of the toxin, and use that in a vaccine. Sometimes only the antigenic molecule itself is given or administered in the vaccine, and we refer to that as a subunit vaccine, when we just give that portion that is the antigen, if we can isolate that from the rest of the antigen-bearing particle. And the goal of a vaccination program is to create what's known as herd immunity. Now, no vaccination program ever gets 100% of the population vaccinated, but the goal is to have, if this is the population here, and all of these are vaccinated, then if we have a couple that are unvaccinated, then the chance of them being sick and being near to another unvaccinated person and spreading the disease is pretty slim. And so, I've heard numbers like if where epidemiologists suggest that if you can get 90% of the population vaccinated, then you've provided herd immunity, because the entire population, the whole herd, is unlikely to, there's not enough susceptible individuals to sustain an outbreak when herd immunity has been created. So that helps to. So the goal of a vaccination program then is to reduce pathogen transmission, and in that way protect the population. Okay, so that's active immunity, both naturally acquired and induced. But we can also offer someone what's referred to as passive immunity. So passive immunity, as the name suggests, does not require a person to actually encounter the antigen-bearing particle in order to get immunity. Instead, we can isolate antibodies from one individual, or maybe from an animal in which the antibodies are grown. So we can isolate them and then deliver them in the form of a shot, and so, or some other way. So there, again, are naturally acquired passive immunity. And about the only example I can think of that is when a mother passes on antibodies either across the placenta, like we talked about with IgG, or in breast milk. So some of them can be passed easily in breast milk. And in fact, the first day or two's secretions that come from the breast following delivering of a baby is actually not milk, but a material called colostrum, which is very, very high in antibodies. And so that delivers then to the baby the antibodies that will provide immunity. And so as long as baby is nursing, the baby's going to have access to all of mother's antibodies as well. And that is naturally acquired passive immunity. Induced passive immunity is when antibodies are collected from, say, the survivors of an infection. So if you remember back last year when the Ebola virus was going on, some American doctors and healthcare workers got infected with it, and they brought them back to Atlanta, to the CDC there, and put them in a hospital and were treating them. And one of the treatments that they used was to isolate antibodies from survivors of the Ebola virus in order to administer those antibodies to these healthcare workers who had contracted the disease. Another example would be the antibodies that are given to fight a rabies infection, or the RhoGAM, the gamma globulin injections that pregnant women are given if they are Rh negative and their husbands are Rh positive, and there's concern that the baby may be Rh positive as well. So we can induce passive immunity by giving antibodies as an injection, or we can naturally acquire passive immunity through nursing or by receiving the antibodies across the placenta. All right, so you know how to reach me if you have any questions, and study well.